If we discover something cool in science, we end up in a textbook somewhere that's obsolete in 10 years. If we discover something that helps a patient, it lives on in the patients having improved lives. That's the reason why we do what we do. I'm Stormy Chamberlain. I'm an associate professor at the University of Connecticut School of Medicine. I work in the Department of Genetics and Genome Sciences. My lab studies chromosome 15q imprinting disorders. And so right now our efforts are focused primarily on Angelman syndrome. For all of our chromosomes, we have one copy that was inherited from mom and one copy that was inherited from dad. It usually doesn't matter, they're both evenly used. For chromosome 15q, that's not the case. For Angelman syndrome, they're missing a protein called UBE3A in their brain cells. To visualize how it works, I like to imagine two train tracks for chromosome 15q. One track represents the paternal copy. The other track represents the maternal copy. At one end of the track, they each have a train ready to make UBE3A. When a brain cell is functioning appropriately, the maternal train travels along the track and makes UBE3A. But on the paternal track, the situation is different. Traveling in the opposite direction is another train called the UBE3A antisense. Eventually, it knocks the other train off the track, and this is why the paternal copy of UBE3A is inactive or shut off. But since the maternal copy is functioning, there isn't an issue in a normally functioning brain cell. However, for patients with Angelman syndrome, they're completely missing their maternal copy of the UBE3A gene. What we need is a way to activate the paternal copy of UBE3A to prevent the train making UBE3A from getting knocked off the track. So because we understand how it gets silenced, we can figure out how to reverse that process. Our solution is to add an shRNA that cuts and derails the antisense train, allowing the paternal train to continue making UBE3A. And so we're going to put back the protein, the exact protein that's missing in kids with Angelman syndrome. Our lab uses induced pluripotent stem cells to study Angelman syndrome. We can take a skin sample or a blood sample from an individual with Angelman syndrome, and we can convert those cells into stem cells. The reason we want those stem cells is stem cells can become any cell type in the human body, so we can convert the stem cell to a neuron. And so in that way, we can study neurons from individuals with Angelman syndrome and test therapeutics in them. They'll plate the neurons on a plastic dish, and in each well of this plastic dish, they will add an shRNA. The shRNA it's nothing more than a set of scissors. It's scissors that we can direct, though, to a very precise place. So maybe we'll have 4 different shRNAs at a couple of different doses. They leave them on those cells for up to a week, and then they'll harvest the RNA. And what we're looking for is the conditions where the antisense is reduced and UBE3A goes up, because that tells us that the shRNA is cutting and that it's turning on dad's copy of UBE3A. The final way we'll do this is we'll try a really big screen where we stick in tens of thousands of sequences. We develop cell lines that let us do it more rapidly. We use CRISPR engineering to allow us to convert them to neurons faster. When we initially started this work, it took us 10 weeks to make a neuron in a dish that we could then test an Angelman therapeutic approach on. We now have it down to a week. That's amazing progress. My people are happy.
Stormy is a great collaborator, and just her talent, her research talent is tremendous. This relationship is really about her knowledge and us leveraging it towards making a therapeutic.
What drives Ovid are the patients. I've kind of watched them be really mindful and thoughtful about what the data and what the clinical trial results means to the patients.
At the end of the day, that's who we want to impact. There's no other reason for us to do what we do. We are dedicated as a company, as a team, as individuals to working with Stormy and the foundations.
We want to get this right. The kids are too wonderful to accept anything less.
I can't think of anything more important, to be honest. We will make this succeed. That's our mentality.
With science, it's not an absolute, it's what we know today. And so based on what we know today, we should be optimistic. I think the whole world should be watching Angelman syndrome from that perspective.